Thank you. We are going to have a course session in molecular biology. As you know, biochemistry totally consists of 16 sessions. Okay. For each session, you will receive 4 MCQ. Okay, each session 4 MCQ. Totally, you will receive 64 questions for the biochemistry part. As you know, blood consists of histology, physiology, and biochemistry. Total together, there are 88 questions, near to 88 like that. But 64 belong to the biochemistry part, and 15, a score of 20, is belong to the biochemistry test. So my part is 4 sessions. 4 multiplied by 4 is 16 questions. So from all of my slides, you will receive 16 questions. Is the clear of actually what we are going to do? The responsible of the course is me, and actually I will finalize your score. Then please try to actually do exam in a good way, in a good manner. If you have any question, you can be in contact with some professor and also with me. My email address is that. Somehow I actually answer my email sooner than Telegram or other actually application. I focus on mail more than. And Gmail actually is more actually updated in comparison to the academic. So is there introducing the course? But what we are going to learn is about the molecular biology or understanding of DNA, RNA, and protein. First of all, we are going to talk about the process actually. Yeah. What is the DNA, guys? Can you give me the definition of?
Two strand of that contain the nucleic information about the genetic material that can build the characteristics of the person growing up and can help in the processes of building the protein.
Great. Well done. It was amazing, actually, definition of DNA. So, what we are as a human, as any organism, as other animal, is based on our DNA. So DNA is the holy book, is the reference, is everything. Everything is based on the DNA. We need to make a copy of DNA sometimes. Why we need to make a DNA? To build? No. To build a protein, we don't need to make a DNA. Yes, we need to make RNA. But why we need to make a DNA? Yeah.
We need to replicate DNA in order, like in mitotic division, in order to get new cells with complete genetic material.
For cell replication, when we are going to make a new cell, we have to provide the genetic material. So why we need to do cell replication? For growth. As you study in embryology, at the beginning we are just one cell. Then we have dividing cells, and then we become an embryo, and then as a human. So we need cell replication for growing, and we need DNA for doing a cell replication. So DNA is so necessary. Process for making a new copy of DNA is known as a replication, because we replicate the DNA from itself. So this process is known as a replication. But as your friend mentioned, what we are is based on the function of protein. When we actually move our hand, we're talking, we're thinking, we're walking, everything that we are doing is based on the function of the protein. The DNA is made from what? Nucleotide. The name of DNA and RNA, they are nucleic acid. It means they are acid in the nucleus. For this reason, we call them nucleic acids. So nucleic acid, DNA and RNA, both of them, they are synthesized from nucleotides. DNA is in the nucleus. Protein mainly are in the cytoplasm. So what actually we can compare that? For example, imagine you have a big reference book in the library. You are going to take some note for yourself. Just those parts are important for final exam. So those parts which they are important for final exam, they are known as a gene. RNA, exactly. So we are going to take some note from the DNA, from the main text, for ourselves. So this process, because we just transcribe from the main text, it's known as a what? Transcription. So the language of the DNA is nucleotide. The language of RNA also is nucleotide. But as I mentioned, based on this instruction which coming from the DNA and then to the RNA, we are going to synthesize the protein. Protein are made from amino acids. So the language of RNA is nucleotide. The language of protein is amino acid. We need doing what? Translation. Because the language is different. Nucleotide to amino acids. So these three processes together, the flow of genetic information from DNA to RNA to protein, totally together known as a central dogma theory. Dogma means is actually stable to the end of life. It's not changeable. We have actually some people, they are dogmatism. Those people, they are dogmatism, they don't change their mind. For this reason, we are saying, please do not be a dogma. Okay? We should actually, but this theory is not changeable. It's the basic of theory of the life. How we are saying in Islam, Quran is the basic of everything, is the basic of everything in the world. Not just in Islam, in other religion, everything. Is the basic of everything in the world. This theory. We are actually at the beginning DNA, then RNA, then protein. Together known as a central dogma, the basic theory of the life, which Francis Crick actually, for the first time, coined these topics. Just we have actually exceptional in some viruses. Instead of DNA, in those viruses, RNA is the main molecule for genetic information. So in those viruses, we have to synthesize DNA from RNA. This process is known as a transcription. This process is known as a making RNA from DNA, making DNA from RNA. We are going to reverse the transcription. So it's known as a reverse transcription. Reverse transcription, because we are going to reverse the process of transcription. So totally together, these processes, they are known as a central dogma theory. The central dogma theory tells us During the replication, we are going to make a copy of DNA. During the transcription, we convert DNA to the RNA, and during the translation, we make protein from the RNA. The flow of information, genetic information from DNA to RNA to protein together is known as the central dogma theory. So, let's go talk about the structure and basic structure of the DNA. So, as I mentioned, DNA is synthesized from nucleotide. Nucleotide itself consists of 3 components. Okay? What they are? 3 components of nucleotide. What they are? What? Nitrogenous base. Then sugar and phosphate. About nitrogenous base, it can be 2 kinds. Purine and pyridine. What is the differences between purine and pyridine? In purine, we have a 2 ring, 1 hexamer and 1 pentamer. Okay? Here we have a nitrogen, 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 and nitrogen. The nomenclature of the carbon and nitrogen is started from that one: 1, 2, 3, 4, 5, 6, 7, 8, and 9. So it's the most common purine. When we are going to make adenine, how many kinds of purine we have? 2. They are what? Adenine and guanine. So for adenine, just we add an amino group here. So the other name of adenine is 6-amino purine. 6-amino purine. So let's have a look at the guanine. In guanine, actually, instead of amino, here we have an oxy. On carbon number 2, we have an amino. What is the name of guanine? 2-amino 6-oxy purine. Just. Let's have a look at the pyrimidine. In pyrimidine, the main structure is from hexamer, 1 ring. This ring has a 2 nitrogen again. The nomenclature is started from that nitrogen. So we have a 3 kind of pyrimidine. Uracil, thymine, and cytosine. Thank you. Uracil is so easy. Just has a 2 oxy group. Okay. So what is the name of the uracil? 2-oxy, 4-oxy, pyrimidine. So if we are going to make a thymine, just we need to add a metal group on carbon number 5. Adenine, 2 and 4-deoxy, 5-methyl, pyrimidine. For making a cytosine, we have to remove this. And then we can add an amino. What is the name of cytosine? 2-oxy, 4-amino, 5. It's the structure of the nucleotide, the most common purine and pyrimidine, the nitrogen space. So in purine we have a adenine and guanine. In pyrimidine we have a uracil, cytosine, and thymine. Then the next part is sugar. Sugar can add to nitrogen number 9 in purine or nitrogen number 1 in pyrimidine. Okay? In sugar, actually, we have a ribose. These ribose, by carbon number 1, it can be added to the nitrogen space. It's 1 prime, 2 prime, 3 prime, 4 prime, and 5. Why we use prime for nomenclature of carbon in the sugar?
It works like a bridge that connects between the phosphate group and the rest of the structure.
Anyone? Any other?
Because there are 5 sugar, 5 carbon.
There are 5 carbon, but why we use prime? 1 prime, 2 prime, 3 prime, like that. To distinguish from the number and the nitrogen space. Here we have 1, 2, 3. Here we have 1 prime, 2 prime, 3 prime, 5. So these two are so important for us. 5 prime, it can be bound to the phosphate, 3 prime, OH. These 3 prime and 5 prime are important for us. So, where?
The carbon number 6, where is it?
Which one is 6? Here
Yes.
is 1, 2, 3, 4, 5, 6. Because I added oxygen, I removed that. And in this one, actually, also it can be added to the. We have 2 kinds of sugar. If on carbon number 2 prime, they have a OH group, is known as a ribose. If doesn't have a oxygen, is known as a deoxyribose. Okay. So when a nitrogen space bind to the sugar together, these two, we are creating a molecule which is known as a what? What? Nucleoside. It's not nucleotide. Nucleoside just consists of base and sugar together. Like that. Like that. So for adenine, is converting the name. Adenine is converting to the what? Adeno what? Side. So adenine convert to the adenosine. Guanine to the guanosine. Uracil to the uridine. Thymine to the thymidine. And cytosine to the cytidine. So when we have an ozyme at the end of the name, it means is a nucleoside of purine. When we have an iodine, it means we have a nucleoside of pyrimidine. Adenosine, guanosine, cytidine, uridine, thymidine.
Professor, can you repeat that?
What?
Can you repeat that?
Yes.
When we have
When-
sign,
Yes, when we have a base and sugar, just base and sugar together, the nitrogen base convert to the nucleoside. Nucleoside is consists of base and sugar. For adenine, convert to the adenosine. For guanine, guanosine. So purine, they have an ozyme at their name, at the end of their name as a suffix. But for pyrimidine, uracil, thymine, and cytosine, they convert to the uridine, thymidine, and cytidine. So they have an iodine at the end of their name.
Understand that point. That's
What?
why we use one thing.
1 prime,
Yes, I understand.
I told you, to distinguish from this has a carbon nitrogen number 1.
Yes.
If we add a 1 prime, it means is different from that. To distinguish between their name. 2, 2 prime, 3, 3 prime, like that. So when adenosine receive a 1 phosphate, is converted to the adenosine monophosphate. Like that. On carbon number 5 is receive a phosphate. When it's received 2 phosphate, convert to the adenosine diphosphate. When has a 3 phosphate, adenosine triphosphate. For guanine is similar. Guanosine monophosphate, guanosine diphosphate, guanosine triphosphate. For uridine is similar. Uridine monophosphate, uridine diphosphate, uridine triphosphate. Also is similar for thymine. Thymine monophosphate, diphosphate, or triphosphate. Also for the site. So when we are saying a nucleotide, we means all kind of these 15 molecules together. A 1 phosphate, 2 phosphate, 3 phosphate doesn't matter. All of them, they are nucleotides. Can you give me the function of nucleotide? What is the main function of nucleotide in our body?
Forming the DNA.
Exactly, making DNA and RNA as a precursor of DNA and RNA. Then, what is another function of nucleotide? Just for making DNA and RNA is necessary.
Saving energy.
As an energy carrier, actually, like ATP, GTP, for example, especially ATP. Then,
Doctor, what is GTP?
what?
GTP, what is GTP?
Or protein, actually.
You
No,
just-
you just told us, but
The
Where can we
guanine
find
and
it?
phosphate. The
where can
guanine.
we find it? I mean,
That's the GTP.
I understand.
Yeah, let me. All of these molecules, they have a similar energy. ATP, GTP, CTP, TTP, UTP. They have similar energy. Sometimes we use ATP as a source of energy. Sometimes we use GTP,
Depends on what?
especially for protein synthesizing. During the protein synthesizing, inshallah, the next week when I teach you the protein synthesizing, you will find out. Mostly we use GTP instead of ATP for protein synthesizing. Is an energy actually carrier. Yeah.
You all get ATP or energy. What difference?
Based on the, actually I told you, GTP also used as energy. But, for example, for making glycogen, we use UDP. For making some of the lipid, we use CDP. Okay? It's different based on what we need inside the cell. But ATP is more common, it's more famous in comparison to others. So we talked about their function. As you actually find out to know, it's important as an energy carrier, as a precursor of RNA and DNA. What? Yeah, but is known as a second messenger. Second messenger in internal signal. Exactly.
Mechanism, because it's carried cofactors.
Exactly. What kind of cofactor?
Coenzymes?
Coenzyme. Okay, not cofactor. Coenzyme.
Yes.
What kind of coenzyme we have? Which they contain nucleotides. Like NAD, FAD, coenzyme A, all of them they contain nucleotide. Flavin, adenine, D-nucleotide. Niacin, amino, D-nucleotide. Coenzyme A, it means actually it contains nucleotide, all of them. If you remember the structure of vitamin and coenzyme, some of them they contain actually these structures.
So, Professor, all the coenzymes.
No, all of them. Because, for example, biotin is not nucleotide. Folic acid somehow is not nucleotide. But NAD, FAD, coenzyme A, they are. So is the three component, as I mentioned, nitrogen space, 5 carbon sugar and phosphate group. In nitrogen space, we have purine and pyrimidine. In purine, we have adenine, 6-aminopurine, guanine, 2-amino-6-oxy, pyrimidine, cytosine, uracil, and thymine. Then we can add a sugar, a ribose or deoxyribose. Deoxyribose in DNA and ribose in adenine. So when we use, for example, DATP, it means we have a deoxy form. DGTP. When we just use ATP, it means ribose form. Okay? So if we add a small d, a small letter d at the beginning of the nucleotide, it means is a deoxyribose. Also, instead of showing ATP, GTP, CTP, and TTP, we can use N. N means it can be all kind of form, A, C, G, or D. When we use NTP, it can be ATP, GTP, CTP, or UTP. When we use DNTP, it means the deoxyribose. Maybe we use NDP or NMP. So N, it can be all kind of four nucleotides.
Doctor, you are using another PDF?
What?
Are you using another PDF?
I sent this PDF to. No,
Ours is different.
no, I sent a new one to your friend. I sent right now to your friend, and actually he sent it to the group. To Ahmed. Just open the file by Ahmed. Send it. It's similar, just a little difference. So deoxyribose anaerobic. What kind of bond there is between the sugar and nitrogen space? What kind of bond? Glycosidic bond. So we have glycosidic bond. Between which carbon? Nitrogen number 9 in purine and carbon number 1 in the ribose. Nitrogen number 1 in the pyrimidine and carbon number 1 in the sugar. So we have adenosine, cytidine, guanosine, uridine, and thymidine. They are nucleoside. They are not nucleotide. Then when we add a phosphate bond, by what kind of bond? What kind of bond here? Phosphoester bond. So here we have phosphoester bond. Here we have a what kind of bond here? Few seconds ago I told you. Glycosidic bond. Here we have glycosidic bond, here we have phosphoester. So adenosine monophosphate, guanosine monophosphate. So as you said, we need nucleotide as an energy carrier like ATP and other nucleoside triphosphate and GTP, CTP, and TTP. For biosynthesizing of, for example, glycogen, lactose, for biosynthesizing of lipid, and for biosynthesizing of protein. They are part of coenzyme, as I mentioned in FAD, NAD, NADP, and coenzyme as a second messenger. Two of them are important, CAMP and CGMP. And they are important for synthesizing of what? DNA. So here you can see the cyclic AMP or CAMP and cyclic GMP or CGMP. CAMP, what's that? When the level of energy inside the cell is high, the amount of ATP is high. When the level of energy is decreasing, ATP convert to the what? ADP. When it's decreasing more, ADP convert to the AMP. Then AMP convert to the what? CAMP. So CAMP is the signal inside the cell to understand the level of energy is so low. So many of hormone, neurotransmitter, and signaling, they use CAMP as a second messenger to send some signal inside the cell. The level of energy actually is low, and you need to provide energy from different sources. Let's have a look at the DNA structure. DNA actually is similar to the state. A state it can be actually linear like that, or it can be actually zigzag like that. Doesn't matter. Most often they are the same. So here you can see the structure of DNA. This structure, it needs two backbone. Two backbone. This backbone actually is
On the nitrogen basis?
What?
It's the backbone is a nitrogen basis?
No.
Nucleus.
Is
Nucleotide
Phosphate
is a
and-
nitrogen basis. يعني مقطع.
is a phosphate and sugar just. Okay, as you know, nucleotide consists of 3 parts. Nitrogen space, sugar and phosphate. Sugar and phosphate are here. So in one part we have a 5' phosphate. In the next one we have a 3' orange. So here is 5' to 3'. The next one is 5' to the 3'. What does that mean? It's anti-parallel. Parallel, it can be here like that, is parallel. Look at my finger. But here is anti-parallel. Parallel, anti-parallel. The DNA is anti-parallel. One side we have 5', 3'. 5', 3'. Look at that, or look at that. So DNA is double-stranded or duplex of anti-parallel, which is a complement. What does that mean, complement? So here, in addition to sugar and phosphate, also we have a nitrogen space. So this nitrogen space,
Connected to each other. One
Exactly.
is connect the other.
they connect from one strand to the other strand. By what kind of bond?
Hydrogen.
Hydrogen. So if you imagine the DNA structure is a state, the backbone is a phosphate and sugar, and the connecting is a nitrogen space. Adenine, thymine, guanine, cytosine, cytosine, guanine, thymine, adenine, like that. So when the DNA is turned around actually axis, some space actually created here, 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 and there. It's like a valley. Okay? Valley means exactly in Arabic. Wadi. Exactly. Wadi it means desert. I think in Arabic.
Wadi? No.
Wadi it means desert.
Space between two
Valley.
Okay, valley it means actually
It's
wadi.
like the area between two mountains.
But I told it myself in Arabic it means desert wadi.
Deserts in
I
Sahara.
mean I know about the Sahara.
Body.
Wadi also.
Body.
Wadi it means like a village. Let's continue. Doesn't matter. This valley also has another name, groove. Groove. We have a two valley. The major groove, the minor groove. Okay, is creating during the creating of helix of DNA. These major and minor are important because they provide some space, exposed bases actually to actually some protein which they are known as a transcription factor bind to these exposed bases, and so important for regulation of gene expression.
Doctor, you said they use hydrogen bond.
Yeah.
Hydrogen bond is a weak bond. Why do they use it?
Because actually it's not a weakened bond, it's weakened in comparison to the covalent. But why? It's a good question. DNA is double-stranded. When we are going to make a new copy of DNA, we have to separate them. Okay? If we cannot separate them, we cannot make a new copy of DNA. So if they bond to each other tightly, it's not easy to separate them. So if other way, if they connect to each other with so weak, so they are not creating a stable structure. So the bond, it should be between the so weak and so strong. The hydrogen bond is a good bond here. And we have a 2 hydrogen bond between adenine and thymine, and 3 hydrogen bond between guanine and cytosine. So those places in DNA which contain a high content of guanine and cytosine is so more actually stronger, is not actually separatable. In comparison to those parts, they have a high amount of AT, because AT has a less hydrogen bond.
Doctor, I have another question.
Yeah.
Which part carries the gene? The base or the sugar or what?
What do you mean?
The genes, I mean.
The gene is a segment of DNA, segment of nucleotide together. Nucleotide means base, sugar and phosphate together.
I mean, when hard
No, let me to actually explain. The gene is a segment of DNA, the whole length of DNA. It's contains sugar, phosphate, and nitrogenous base together. Maybe 1,000, maybe 2 million, maybe 10,000. All of these nucleotides together known as a gene. Okay? We do not separate the... So, but for showing the sequence, we just use the nitrogenous base, adenine, guanine, cytosine, like that. So, this DNA turned around actually axis, as I mentioned. This is the axis. We have a 2 number. The number doesn't matter totally, but the space which actually cover the 2 nucleotides is about 3.4 angstrom. In each turn, actually, turn means when the DNA turns 1 time. In each turn, we have a 10 power to the 5 nucleotide together. So totally, in a 1 turn, the space is near to 34x in one term because we have a 10 nitrogen space, 10 base pair.
Should we remember these numbers?
No, I told you. But in the next slide, we have some number you need. We want to change the color. So base pairing, it means nitrogen space in one strand is binded to the next one, in the next phase. Two polynucleotide chains in the double stranded are connected by hydrogen bond. As I mentioned, they are weak in comparison to covalent, but they are not so weak. We have a rule which is known as a Chargaff rule. Did you hear about the Chargaff rule?
Yes, like
Always adenine bind to the thymine and guanine bind to the cytosine. So the amount of adenine in the double stranded DNA is equal to the time, and amount of the guanine is equal to the amount of cytosine. So imagine we have a segment of double stranded DNA, and in this DNA we have a 10% guanine. How many percentage we have thymine?
No,
How many percentage?
it's not related.
90.
It's not what?
90.
90?
90,
90?
90.
90, he says.
90.
But it's not related because guanine is not related to the thymine.
We don't.
how we can know?
No.
We can't collect it.
You can't?
You can't collect it because guanine is not related to thymine.
Okay.
So if you want to cytosine, it will be 10%.
So if we have a guanine, how much cytosine we have?
10%.
10%. Okay. So the total of the DNA is 100%. When we have a 20% of them, how many adenine and thymine we have? 80%. So they are equal. 80% divided by 2, how much?
40 for each one.
So it's calculable. Okay.
Okay.
You got it? So it's not so difficult. If we have a 10% guanine, we have 40% thymine or adenine. Because we have 10% cytosine, 80% is remain divided by 2 is. So if we have a molecule, RNA molecule actually, it consists of 20% actually adenine. How many percentage we have cytosine?
40.
30. Any other idea?
Yeah.
It's a tricky
30.
question.
30.
Why is it a tricky question? Because I told you we have RNA. I don't tell you we have DNA.
Oh.
So RNA is one single stranded. So in the single stranded we cannot calculate. It can be different. The amount of adenine, thymine, cytosine, guanine, it can be any number. We cannot calculate. Just in double stranded DNA is calculable. So it's about the Chargaff rule. And as you saw, always thymine bound to the adenine by how much hydrogen bond? 2 hydrogen bond. Guanine and cytosine? 3. So it's so easy. The charge of DNA is negative or positive? Negative. Why? Because of phosphate. Phosphate always has a negative charge. So for having a phosphate group, DNA is a highly negative charge molecule. Remember that? Then we will actually work with that here. As you saw, is antiparallel 5' 3' OH. 5' phosphate, 3' OH. We have a term which is known as a genome. Can you give me the definition of genome?
It's a section of DNA.
Is genome, not gene. Gene is a section of DNA, but genome. What is a genome? Yeah?
DNA and like a histone.
No.
Like whole genetic materials
Whole genetic material
and the DNA.
in where?
Is it organic or what?
In one cell. Whole genetic DNA, whole chromosome. In one cell known as a genome. Because all of our cells, they have a similar DNA, so the genome is similar in all of our cells. So genome, it means the total DNA content inside the cell. Those is actually field which they study genome, they are known as the genomics. The genomics is a field of study of genome. In comparison to genome, we have another term, like transcriptome. Transcriptome means the total amount of RNA inside the cell. And the field of study is transcriptomics. We have another term, proteome.
Total amount of protein.
The total amount of protein. The study is known as a proteomics. Also, we have a metabolome. Metabolome means
Total amount of
metabolite. And the studies is metabolomics. Till now, the scientists, they were successful to study the genome, and genomics is completed. Because we know the all genome inside our cell. But about the transcriptome, proteome, and metabolome, they are studying and researching. Maybe in the future, we'll be able to talk about the transcriptome, proteome, and metabolome. So, tell me, what is the amount of genome inside our cell?
3 billion.
3 billion, thank you. So, we have a 3 billion base pair, or 6 billion base, doesn't matter. If you are using double-stranded base pair, in one strand, actually is 6 billion. So, what is the length of these 6 billion base pair? The length. The length. Masafa. Can we calculate the length of DNA inside the cell?
You mean length? Length?
Length, yeah.
We cannot.
Which one?
We cannot?
I don't think so.
Why?
Because it's inside the nucleus.
No, we can't. Because I told you, the distance between two nucleotides is 0.34 angstrom. You can multiply them. So if you multiply that, A base pair is equal to the near to 1 meter. A double stranded is equal to the 2 meter. 1 meter DNA or 2 meter, doesn't matter. Do you have any idea about the cell size? What is the cell size in our body? Yeah. How much? 1 nanometer.
Doesn't it depend on the function of the cell?
Yeah, but totally. 1 nanometer is not possible actually. It's less than a molecule. Yeah?
I'm not sure, but like maybe in micrometers.
Exactly. It's completely correct. But how much?
1?
No, 1 is so
10?
Yeah. The one of the most smallest cell in our body is RBC, which is 7 micrometer. Okay? But most of our cells, they are between 10 to 30 to 50 like that micrometer. The largest cell in our body is ovum. Ovum is about 200 micrometers. Okay? Is the largest one. But the normal range is that. So, is a micron is a meter. Meter to micron is more than 1,000 times. So, we have a DNA, 1 meter, which is 1,000 times, is bigger than the cell, which is 10 micrometer. It's not possible to show you, because just under microscope you can find a cell, as you saw in the histology and embryology. Okay? Just by microscope we can see the cell. It's actually, but 1 meter is that amount. How is possible that amount of DNA, 2 meter or 1 meter, goes to the...
Because it's condensed.
Yes, exactly. Like that. Thank you. By packaging, compacting. So we need to compact DNA. How we can compact DNA? DNA charge is negative or positive? Negative. So we need a protein for compacting.
Negative protein?
What kind of protein?
Histone.
Histone. Histone, they are positive or negative charge? They have a DNA has negative charge. Protein should have a positive charge. So they should have a basic amino acid.
Doesn't each DNA connect to 4 molecules of histone?
Yeah, let's me to explain about the histone. Just first of all, the histone actually, they consist of basic amino acid. What kind of basic amino acid you know? Yeah.
Lysine.
Lysine?
And
No, lysine is not. No, tyrosine is not.
What?
Basic amino acid. We have 3 basic amino acids. One of them is lysine.
choline.
No,
Pyruvic?
no. Pyruvic is not amino acid.
Pyruvic.
Proline, but what is not? The second one is arginine.
Serine?
No, serine also is not basic. Histidine. Histidine, arginine, and lysine. They are the 3 basic amino acids. So in histone, we have high amount of lysine and also arginine. For this reason, histone, they have a positive charge. So 5 minutes less than that. DNA structure. DNA, as we saw, is a polymer of deoxyribonucleic acid, actually, which they are linked covalently 3' to 5' phosphodiester bond. So phosphodiester bond actually is connecting this backbone for us. Just in some viruses, actually, we have a single-stranded DNA. All other organisms, they have a what? Double stranded DNA. Just in some viruses, we have a single stranded DNA. The other organism, all of them, they have double stranded DNA.
What's the name of the virus?
What kind of virus? Right now, I don't remember. For example, some herpes viruses, they have one. You will study in the actually first. I'm talking about that single stranded DNA, ssDNA, DS double stranded DNA. Yeah. Some viruses, actually, your friend asking me about the name of viruses, I'm not sure about it. But herpes simplex viruses, actually, hepatitis B, they are actually DNA viruses. Some viruses they have RNA, some they have T. Virus is kind of organism. They consist of nucleic acid and protein, but they cannot do the replication. They need a host. When they are coming to our cell, they can.
I think you say virus, not virus.
They're similar. What is the difference? The correct pronunciation is virus. It's not virus. It's virus, actually. For this reason, I pronounce that. Doesn't matter, but totally you understand what I'm saying. So it's about the group or valley in the DNA, actually. They have two functions. First off, they are site for binding of regulatory protein. Regulatory protein. The second, this part is so important for understanding what's happening. For cell replication, actually, we have two kinds of cell, eukaryotic, prokaryotic. Okay? Eukaryotic cell and prokaryotic cell. Eukaryotic cell, it means our cell. Those organisms, yes, they consist of many cells. Prokaryotic, they are single cell, like micro. Okay? Is the difference. First. Then, prokaryotic and eukaryotic, for cell replication, they need DNA and RNA and protein. So, if we inhibit synthesis of DNA or RNA or protein, if we inhibit the synthesis of these three molecules inside the prokaryotic, those drugs, they are known as antibiotic or antibacterial or anti-infection drug. So most of well-known antibacterial agents, they are the inhibitor of DNA synthesis, RNA synthesis, or protein synthesis.
For the harmful bacteria.
So,
For the harmful bacteria.
for all kinds of bacteria, they can be. For this reason, when you use antibiotics,
You can't use too much.
they can also
Kill the
degrade
bacteria.
the useful bacteria. If we inhibit DNA synthesis, RNA synthesis, or protein synthesis in eukaryotic cell, it can be used for what kind of disease? Anti-cancer. So many of anti-cancer drugs and many of antibiotic drugs, they have an effect on these three molecules: DNA synthesis, protein synthesis, or RNA synthesis. During my session, I will give you the name of some of them, not all of them. But in the next years, you will study a lot of them. So if we inhibit each of these processes in eukaryotic is anti-cancer, in prokaryotic is antibiotics. When we are going to make an anti-cancer drug, the the sequences of the group is so important. So when we are going to design a drug as an anti-cancer, it's so important to have information of them. One of the most of anti-cancer, actually, which is so famous, is actinomycin D. Actinomycin D bind to here, for example, and doesn't allow the transcription is active. So is the function of the group for regulatory protein binding and for designing an anti-cancer drug.
It's the same of the chemotherapy? Or
Yes, chemotherapy is when we use the anti-cancer drug is a chemotherapy. We have three forms of DNA inside our cell: aDNA, bDNA, and zDNA. The most common and typical form is bDNA. 95–99% of DNA in our body is bDNA. Just in some specific area or condition, we can have an A or Z DNA. During the, for example, DNA is dehydrated, the aDNA is created. When the level of water decreases inside the cell, the aDNA may be created. And when we have a high AT amount, the repetitive of 80, we have a set. If you look at their structure, the B1 is the main one and is the normal. How many base pair we have inside each term is 10.5. And if you look at that, is a right-handed. Right-handed means, look at my finger, for me is right-handed, for you is left-handed, my finger. But vice versa, for me is left-handed, for you is right-handed. So bDNA and aDNA, both of them, they are right-handed. They are turning right side. But zDNA or zDNA is left-handed. So here we have 11 base pair, and here we have a 12 base pair. Here is more taller, here is more shorter. Is the normal. Just is the differences between aDNA, bDNA, and zDNA in our body.
Doctor,
Yeah?
the form of DNA is defect or not? The human body.
Yeah, but you know, totally we don't have aDNA and zDNA. Just when, for example, inside the nucleus is dehydrated, aDNA is created. Doesn't have any other effect actually. If the DNA completely converts to aDNA or zDNA, yes, it has a dangerous effect. But totally we don't have that.
Doctor, why is the DNA more stable than RNA?
It's a good question. Why DNA is more stable than the RNA?
because it consists of two.
One of the reasons is that because it's double stranded in comparison to the RNA, which is single stranded. Then
Does the uracil in RNA?
uracil in RNA, no, is not the main reason. One of the other reasons because of rare ribose and deoxyribose. This oxygen in the ribose is more reactive. When we don't have oxygen, it's not reactive, so it's more stable. There are many other reasons, but these two are the main ones. So we have another term, denaturation and renaturation. Denaturation and renaturation. What does that mean? Denaturation means when we opening the DNA, two strands of DNA is separated. It means denaturated. When again they rejoin is renaturated. So when we can open the DNA by cutting the hydrogen bond. So in a temperature, for example, the DNA is separated. This separation known as a melting. The temperature of melting known as a TM, melting temperature. Because between adenine and thymine we have a 2 hydrogen bond, and between guanine and cytosine we have a 3 hydrogen bond. If DNA content high amount of GC, high GC content DNA, the TM is increased or decreased? Increased. Because we need to give more energy to separate 3 hydrogen bonds. But when we have a high AT amount, the melting temperature is decreasing. Because we need to just separate 2 hydrogen bonds. So TM is based on the GC amount or AT amount. GC amount increase the TM, AT amount, actually AT reach decrease the T. So the definition, TM is referred to the temperature that the double-stranded DNA is denatured, dependent on the GC content or GC percent. Also pH is important, but GC is more important for us. So we talked about the primary, secondary, then we are going to talk about the quaternary. DNA in the eukaryotic is linear, but in the prokaryotic is circular. When it's circular, actually, it can be turned around itself and can create a superhelix structure. Superhelix structure like that. I don't know you actually had this phone which they have a wire or no? The classic phone, not mobile.
Yes, telephone.
Yeah, those phones they are classic. They have a wire. This wire is a helix. Okay? When you put the phone, what's happening for the wire? It's turn around itself again. It's create a shape like that. So this shape known as a superhelix, and this one is a helix. So when the DNA actually is circular, a superhelix is created actually. Supercoil also is known as a supercoil. Supercoil, superhelix, super twist, all of them are easy. So let's have a talk about the packaging of DNA. We talked about the actually histone. Histone actually are octamer of histone. Octamer means 8 histone. 8 histone consists of 4 kind of histone: H2A, H2B, H3, and H4. So, from each of them we have a 2. This is structure. 8 of these histone together, they create a bead similar to that. Is a bead. Okay? Is an octamer. So this is our bead. Also we have a DNA. DNA as a strand. But here in the a strand pass through the bead. Okay? But in the DNA, wrap around it like that. Wrap around it. So imagine is the structure of DNA. But instead of passing through that, is wrapped around all of them. So these structures, they are known as a nucleosome. Nucleosome. Okay? When histone actually wrapped by 2 times of double-stranded DNA, which they are known as a nucleosome. Also, between these two, for example, imagine there are two bits. There is a space between these two. This space. This space is covering by, for example, that one, which is different from the others. In SEPA also we have some another shape to separate 33 from each others. But between each two nucleosomes we have one of them, which is covering by histone H1. This segment of DNA known as a linker, which covering by histone H1. What is the function of histone H1? Histone H1 actually take this nucleosome and this one and lead to the more packaging. But totally what's happening? Imagine it's DNA and nucleosome. When we are going to pack that, first of all we actually wrapped one time, fold, second time fold, fourth time fold, and finally actually it's going to be so smaller. Then we can put it, and the chromosome is similar to that structure. How much we need to actually compact? More than 1,500 times DNA is compacting to create a chromosome, to create that shape which we have in chromatin or chromosomes. And the function of histone is so important in compacting and packaging of DNA. Is the role and the function. So the structure of DNA is finished right now. Then we are going to talk about the replication. The second file, which I'm ascended to the DNA replication. Replication means making a new copy of DNA. Thank you. So imagine you graduated, you have your office, you make money for yourself, and you are rich right now. You are going to make a building for yourself, okay? Not buying. You are going to make those favorite your building. So what you need? Blocks, building blocks, yes. Then actually all of these building blocks
Basic
are
land.
material together.
Basic land.
Land, okay. Earth, yeah. Then
A map for building. For a map for the shape.
map. Also we need map, yeah. What? They are material actually. They belong to the material. Builder. Builder means worker. Yes, we need workers. Then money, energy. Yes, exactly. We need money. Then land, plan, land and plan, both of them. Plan or map. Yes, we need plan or map. Then we need what?
Workers.
Workers. Actually, your friend mentioned that. Workers, yeah. Start to building. What do you mean exactly? What was that? What do you mean from the signal?
Like you say you should start building.
Okay, what's that exactly? In building is license. Roxanne, license. You need to have a license to go to make a building. Yeah. Money, yes. Exactly. We need supervisor, engineer, actually, to recheck everything. So all of them you mentioned. Thank you. Exactly, it's similar to what we need for making a new copy of DNA. When you are going to make a new copy of DNA, we need worker. They are enzyme.
DNA polymers.
We need, yes, DNA polymerase is one of them.
Not the RNA.
We need actually building blocks. They are ATP, GTP, TTP, CTP. We need energy. Again, these ATP, GTP, they are energy. We need supervisor. They are enzyme themselves. They are active.
map, which is the DNA.
We need map, is DNA itself. We need license. License is known as the origin of replication, the site of starting of replication. We don't have a law to start from any site that we love. For example, you cannot go outside if you saw free space, make an apartment for yourself. It's not possible. They give you the license for that place specifically. You have to do it on that specific place. So all of them, they are similar to making a new copy of DNA, and we have to learn which part is more important for us. First of all, actually, we have a 3 rule in making a new copy of DNA. What are these 3 rules? First one, always DNA synthesizing is semi-conservative. Semi-conservative. What does that mean?
Like, one strain is completely new.
Yeah?
The other one is like from the old.
Exactly. But if we are going to explain that, imagine we have 2 old strands. They are known as the old strand. Old strand or mother strand or father strand or parent strand. When we are going to make a new one, these 2 green, they are known as a new strand or daughter strand. Why we use daughter? We don't use son.
It's similar to Alexa.
What is the question?
Sons are useless.
Why we don't use sons? We use daughters. When they become, actually, we can tell them father strand or parent strand. But when we make a new one, at the beginning, it's known as a daughter strand.
Because we cut double strand to 2 parts, and RNA repeat 1 part.
No, the answer is so easy because it's going to be a model for the new strand. Again, we use it for the making a new.
Sorry, I said that.
So it's like,
You said that? I didn't get sorry.
like we know
Yeah,
that-
because it can be a model for the new strand. For this reason, we use it a daughter strand.
we know that it's a female.
Yeah. So, why we are going to do some replication, DNA replication for cell replication? So when we have a cell replication, we're making a new cell from the previous cell. There is many kind of replication. One of them is conservative. Conservative, it means we keep, actually, let's need to change that one. One type of replication is conservative replication. Conservative means keep the old strand for itself, give the new one to the new cell. Why we are not doing that? Yes. How we actually do by this way. Imagine is the previous one, is the new one. In the previous one, we just have one old strand. In the new one, we have one old strand. And also we have one new strand. Is that semi-conservative? Is that conservative?
Because we need to know like difference. no?
Because each one is complementary for each other.
Your complement, yes, but doesn't matter. These 2 are complement, these 2 are also are complement.
Yeah, but like the green, like both of them, like for example.
No, these 2 also are complement. Doesn't matter for the complement.
Professor, if we're talking about the division and the fertilization, then we need genetics from both the parents, so we take a strand from each one. to make a full collection of genetics.
Because we need mixing the mixing
Yes,
the
mixing of the
material,
genes.
genetic material.
Because the new depend on the old.
Is new depend on the old. Why we don't give the new one to the new cell? We keep one of the oldest strand in the new cell.
Maybe there will be like some.
Because the new cannot work into the old.
Because
No, it can work.
If it's completely new, it may like carry other genetic problems or stuff. So
Oh.
we need like old one.
Carry
Exactly.
the very piece of information.
Is the correct answer, actually. What was the answer? During the replication, maybe we do some mistake. Okay? Some error or mistake is happening. So if we give this new strand to the one cell, we have a mistake here. And we don't have
The old one.
the old one, the main one, the reference to fix that. So we cannot fix that mutation. The mutation is remain, and actually these mutations are increasing. But here, if we have mutation, or here, because we have the main, actually the text, we can correct that based on the main text. For this reason, always it keeps one old strand and gives one new strand. Because if we did a mistake, we can fix that. Otherwise, there is no way to fix that problem. So it's the first rule of replication. The second one. The second one, always, the replication is 5' to 3'. So always we synthesizing from 5' to 3'. What does that mean? It means if this strand is started from 3' to 5', we can synthesize from that side to that side. It means synthesize 5' to 3'. But if this strand is 5' to 3', we cannot synthesize from that side.
Why?
Because it's not possible, it's our rule. It's our rule. So it's a big problem during the replication. In the next slide, you have to tell me how we can fix that problem. For the one strand, we don't have problem. For the second one, we have many problems. The last rule is the synthesis of DNA, synthesizing of DNA always is bidirectional. What is the meaning of bidirectional? Two direction. When it started from one side, is going from two direction actually. Finally, they reach to each other. Or actually, is if linear, the direction is actually one replication from that side, one replication from that side. So we have these three rules, which they are stable. Actually, they are fixed. Semi-conservative, bidirectional, 5' to 3'.
The second, I don't understand it. If you can repeat it.
Also, I didn't understand that myself. It's so difficult for understanding that. Let's, in the next slide, when we actually going to talk about the replication, you can understand what's happening. So, we have a history in Farsi. Actually, there are two writers in the past, not right now. They are saying to each other, I just take, for example, 10 tumor when I'm writing something for someone. And I'm receiving 10 tumor for reading that for that person. The other one just saying, just I can receive 10 tumor for writing because I can write, I can read my handwriting also. Just I can write something, I can read it for myself also. For this reason, I can just tell you something, I can read it for myself again. I don't know, you got it, what was the history or no totally. I explained in a bad way. So this one is a semi-conservative. Always we have a one old strand and one new strand. And it's about the actually bidirectional. Always we start from the origin and we go from two sides. Just we have an exception in some viruses. We have a unidirectional. In some viruses, the length of DNA is so low. As you saw in eukaryotic, is for example in human is a 6 billion. In prokaryotic, in bacteria E. coli, is about 4 million. 4 million. In viruses is about 1,000. Like in influenza virus is 3,000. In HIV is 10,000. In coronavirus is 30,000. So they are so actually small. By unidirectional also they can do the replication. So bidirectional replication involves two replication fork which move into opposite direction.
Professor, is this for memorizing?
What?
Is this slide for memorizing?
Yes, this slide is so important for you. I told you at the beginning we need some workers, some protein, some factor. Is those factors? First of all, imagine is the DNA. Okay. In the DNA in the prokaryote, first of all, we have one origin of replication. Origin of replication. Imagine here is the origin of replication. Also, we have some other nucleotide which they bind to each other. This area is origin of replication. First of all, we have to recognize that places. How we can recognize these places? By the protein, the name is what? DNA A. What is the function of DNA A? Recognizing the origin of replication. So these places is recognizing by DNA. Then we need to unwind the two strand. For unwinding of two strand actually, cutting off the two stranded, we need another protein which the name is, or other name is helicase. Helicase unwind the DNA. You don't need to memorize the number of subunit and this one. Also, you don't need to memorize that one, and this one. No, the other part is important. So, DNA C helping DNA B actually to bind to the origin. So DNA B unwind DNA, unwinding the DNA. DNA C helping. When we actually have an unwinding segment, is known as a single-stranded DNA because it's not complement to others. We have some enzyme, the name is nuclease. What is the function of nuclease in our body?
It's mainly responsible for transporting between the interior of the cell and external space.
What is the function totally? Nuclease. They are degrading the
Involved.
nuclease,
Nucleic involved.
nucleic acid, like DNA and RNA. They are degrading DNA and RNA. They are known as DNAs and RNAs. So, nuclease, they are digesting the RNA and DNA. We have two kinds of nuclease, endonuclease and exonuclease. Endonuclease, it means they cut DNA at the middle of the strand. They cut DNA at the middle. Exonuclease, they cut DNA from the end of DNA. So themselves they can be divided to two kinds: 3' exonucleases, 5' exonucleases. When the DNA is opening and convert to the single stranded DNA, these nucleases they can digest the DNA. Also, when we separate DNA, these two strands, they have tendency to rejoin to each other. So they have tendency to rejoin. We don't want to, they connect to each other again. We have to prevent them. How we can prevent them? Look at my hand. My hand are two strands. I separated them, but they like to rejoin. How I can?
Inserting an enzyme.
Exactly. With binding protein, I covering my hand by some protein, so I can allow them. Here we add some protein actually, which they are known as a single stranded DNA binding protein. SSBP, single stranded binding protein. They have two functions. One of them, prevent from reconnection. The second one is protecting against nucleases.
Protecting from what?
Nuclease.
Nucleus.
Also, we have another enzyme, DNA gyrase. Which one of you can explain for me? When, imagine we have a two strand, these two strands are wrapped around each other. When we start to open that, what's happening?
It's be like this. This will be structure.
It will not will form.
Imagine these two strands are actually wrapped around each other. At the beginning, we can't open them easily. But when we open them, what's happening in the front? Tie is created.
Knot, yes.
Okay? What's that?
Knot. A knot. It's
Exactly.
like a tie.
Tie. A lot of tie is created.
The binding soil.
So this DNA polymerase is unwinding and going, but in the front the tie is created. You got it? You didn't got? Can you explain for them?
من نربطها بينسوية من نفتحهم بالاخير راح يصير اكو عقده بالاخير ربطه تربط فهو اللي يقول انه ليش بعدين راح يصير اكو عقده بالاخير.
Okay. Both them means tie. Okay. Like that is tie. The tie is created. So we have to relax that tie. How we can relax this tie?
By this enzyme?
Yeah. How we can? For example, imagine this is the tie. How I can relax that tie? By hand. But sometimes it's not possible by hand. What can I do?
Cut.
Yes, exactly. I should cut it. I should cut it and again relax the DNA and rejoin that. So we have a specific enzyme. The name is DNA gyrase. DNA gyrase or topoisomerase type 2. The function is relaxing of DNA by two kind of reaction. Topoisomerase actually or DNA gyrase has a two side and has a two function. One of them is endonuclease, which cut the DNA. Cut. The other side or arm is sealing or has a ligase activity. So nuclease activity, ligase activity. Cutting the DNA, sealing the DNA. So cut the DNA, relax the DNA, again seal that. So these enzyme is so important.
The final exam.
Yes, also in the final exam. Why? Because most of the drug can inhibit that in prokaryotic and eukaryotic. Ciprofloxacin, nalidixic acid, actually, they are the antibiotics. But if we inhibit that in human, it's as an anti-cancer, like etoposide, doxorubicin, novobiocin. Actually, in the next slide, I have them, the name of them. So if we inhibit the topoisomerase in prokaryotic or eukaryotic, it's a good drug as an antibacterial or anti-cancer drug. So what was the role of single-stranded binding protein? What was the function of single-stranded binding protein?
Protection.
Yeah, protection against?
Mutations.
Exactly, and then?
Single-stranded DNA.
Exactly. Is the two role of single-stranded DNA, single-stranded binding protein? So it's about when you are actually unwinding the DNA, at the front of unwinding, actually the superhelix or supercoiled structure is created. If you imagine you are opening a strand, at the end is actually sealed. At that places, superhelix or supercoil is created. Some anti-cancer agents like etoposide, doxorubicin, novobiocin, they inhibit human topoisomerases. Also, bacterial DNA gyrase, it can be targeted by antibacterial agent which they are known as a quinolone. Quinolone are a big family of drugs, which one of the most famous is known as a ciprofloxacin, which daily is used for treatment of the infection, ciprofloxacin. So these topoisomerases are responsible to relaxing the DNA. They have two functions, endonuclease and what? Ligase. Endonuclease and ligase. We have two classes, class 1, class 2. Class 1, cut one strand, doesn't need ATP. Class 2, cut two strands, needs ATP. Gyrase is a type of class 2. In the process of replication, actually, we have a three main steps. Initiation, elongation, and termination. Initiation or starting. Actually, for starting, we need DNA A to recognize, we need helicase, we need topoisomerase. All of them are needed at the initiation stage. But in the elongation, actually, we are going to synthesize the DNA from the old strand. This is known as a replication bubble or replication fork. Why we are talking a fork or bubble? If you imagine this is my actually double stranded. If I open that, similar to this structure, is a fork actually, and go by two sides. But it can be similar to bubble, not a fork. Most of them are correct. It can be fork or it can be a bubble.
If it's not separated, it can look like a bubble, or if it's separated,
Yeah.
it can look like a fork.
Fork. Replication fork or replication bubble. So it goes from two sides. One direction and the second direction. So let's have a 10-minute rest and I will finish. We have a two-strand chip. One of them is started from 3' to 5', the second one from 5' to 3'. We need DNA polymerase, actually. DNA polymerase. We have three kinds of DNA polymerase in the prokaryotes. DNA polymerase type 1, type 2, and type 3. Okay? The main DNA polymerase in the prokaryotic is DNA polymerase type 3. In the next step, we will find out why DNA polymerase type 3 is so important for us. But if you imagine, here is the template, and we are going to synthesize from 5' to 3'. So the direction is correct, because one of the rules always was that we synthesize in 5' to 3'. But none of DNA polymerase cannot start from the 0 point. Okay, they can't start from the first nucleotide. Always they need a segment, actually. This segment, it should be provided by RNA polymerase, which the name is a primase. Primase or DNA G. Another actually component of that table, DNA G or primase. DNA G or primase synthesizing a primer. Primer is a segment of RNA, actually. Maybe 10 nucleotide to 30 nucleotide. Is a segment of RNA, 10 to 30 nucleotide. What is the function? Is providing the OH group, a free 3' OH group, for DNA polymerase to synthesizing a copy of DNA. So because the direction is 5' to 3', DNA polymerase type 3 doesn't have any problem, just need a primer, which primer by primase is provided. And synthesizing is continuously is synthesizing. The name of this strand known as a leading strand. Leading strand. قائد strand. Okay? But in the next strand, what's happening? The direction is 5' to 3'. Is it possible to synthesize in 3' to 5'?
No.
No, it's not possible.
So should I?
because it was the rule. I told you 3 rules we have. Always. One of them was synthesizing 5' to 3'. This is the rule. DNA polymerase, they can synthesize just from 5' direction to 3' direction. And they need a 3' origin. Yeah.
So we should skip this part and linkage together by details. So
How we can, first of all, we have to synthesize them.
we don't need to synthesize right now. We should skip it. Then we like linkage together by details.
We don't need to synthesize the other strand?
No, this part we can't skip it.
No, it's not possible. If we skip one nucleotide, also lead to the mutation.
We should link it by when they finish. We should link it by details.
Actually, we don't have any strand. What we linkage? First of all, we should have something, then we linkage them. We don't have anything here. How we can synthesize that?
Let's go from this direction, opposite direction.
Opposite direction is not possible in the opposite direction. Pay attention here, just I'm explaining one time. I hope you understand. If you didn't understand, it's your problem. Imagine this is the bottom strand here. The direction, actually, I told you, we cannot synthesize from this side to this one. So what we can do, we select the segment of that strand. We turn it around and bring it. Here is we can synthesize from 5' to 3'. But we cannot go to the end and synthesize. Instead of that, we bring that to here. Okay? Then we synthesize that segment and it will be finished. The upstream actually strand is coming to us. Then we reach to that part. Again, we create a loop, we synthesize that segment, and we go to the next one. One by one is created. This segment, they are known as an Okazaki fragment. You got it?
So instead of synthesizing a long chain,
Yeah.
you just divide it into 3 chains that one over the other and synthesize it. Then get one over the other and synthesize it.
Exactly. We need a primer here, first of all. Then we create a loop. Then we synthesize that segment.
After that, another
5' to
primer,
3'. Then we go to the next. Again, we have a primer,
make a loop and synthesize it.
synthesize that. Again, we have primer,
Make a loop.
make a loop, and synthesize. Segment by segment. So the synthesis of each strand is this continuously. So this strand known as a lagging strand. Lagging strand. What is the meaning? Is mean follower. Follower of actually leading. So you got it, what's happening here? This actually segment, they are known as Okazaki. Why Okazaki? He's a Japanese scientist. Okazaki actually, with her spouse actually, Tsunake Okazaki and Shinzo Okazaki, they worked together and finally they introduced this kind of synthesizer. But unfortunately, Okazaki, the Shinzo, because of leukemia, passed away at 40 years. And he was born during the actually Hiroshima atomic bombard. For this reason, he had a leukemia. I will actually give this example to a student. Sometimes we have a lot of problems. Like Okazaki had a leukemia from when he was so young. But he didn't actually escape. He didn't stop everything. He worked, and right now we have a segment in DNA as an Okazaki name, and his name is alive to the end of the universe. To the end of the universe and also to this. So, we have a two-part, leading strand, lagging strand. Here is continuous, here is discontinuous. So leading strand, synthesize is continuously. But lagging strand is discontinuously, segment by segment. RNA primer, as I mentioned, DNA polymerase, all of them, cannot initiate synthesis at the beginning. So they need another enzyme primase which provide a segment of RNA. This enzyme known as a primase or DNA G. What is the function? Synthesizing a short segment of RNA, 10 to 30 nucleotides. Then we can start, and initiation is finished. Here, this figure actually is so good to our understanding what's happening. This is the double-stranded DNA is unwinding. What is the unwinder? DNA helices, unwinded to DNA. So because in the front of unwinding we have a super helix, we need what kind of enzyme? Topoisomerases. Topoisomerases. So the synthesis of this strand, which known as the leading strand, is continuously. Just we have one primer and 5' to 3' we synthesize that. But in the next strand, we synthesize them as an Okazaki fragment. Each Okazaki fragment has an RNA primer and a segment of DNA. So it's the whole process of the actually synthesizing of DNA. It's not the correct image of the Okazaki. I made a mistake. It's actually Hollywood actors. I find that the structure is different actually. Let's bring that for you. This one is the Shinzo Okazaki and his wife, Tetsunoka Okazaki, which still is alive. Tetsunoka Okazaki is alive right now. And this image is for 2 or 3 years ago. Reiji Okazaki, which was born in Hiroshima on 1930. He passed away on 1975 due to the leukemia. Have a look at these processes.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork.
So it was helicase, unzipping, cut up. Repeat what? This part.
Separate the two strands. A prime end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process.
So these nucleotide are DNA nucleotide or RNA nucleotide? RNA, because primase is RNA polymerase.
This enzyme makes a small piece of RNA called a primer.
So just a small piece, maybe 10 or 30 nucleotides.
Construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA.
What kind of DNA polymerase? Improprietary. Type 3.
A polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA.
So it's going to talk about the next step. Because these RNA should not be present in the DNA, we have to remove that. Okay? Because they are RNA. We need another enzyme. The name is DNA polymerase type 1. DNA polymerase type 1. By endonuclease and exonuclease activity, actually remove the primer and replace the nucleotide by the what? DNA nucleotide instead of RNA. Then what's happening? Imagine that is the next strand here. I am DNA polymerase type 1, removing RNA, replacing by DNA, till I reach to that place. Because just I can add a nucleotide, here I cannot add a new nucleotide. What's remaining here? A gap. So there is a gap between this segment and that segment. So we have to join them, to seal them. We need another enzyme, the ligase name.
Primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA.
So the gap is remained, the knife here.
Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand.
Seals the
DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Doctor,
Yeah.
molecules need energy to do this process. So how get the energy? How they get the energy?
ATP, GTP, CTP, TTP, they are high energy compound in our body. These molecules, yeah.
The start of hobbies. We have some specific
The start of what?
we have a specific code to start.
We have a specific, yeah, origin of application. This origin of application is so important. So leading strand and lagging strand and the differences I told you. DNA polymerase 1 actually remove the primer, fill the gap, and finally DNA ligase seal these two segments by creating a phosphoester bond between these two nitrogen space and actually fill the gap. So DNA ligase in eukaryotic use ATP as a source of energy, but in prokaryotic use NAD as a source of energy. DNA polymerase type 1 is a very important molecule which discovered by Arthur Kornberg in 1955, and he received a Nobel Prize just for discovering this molecule. We have another term, the name is proofreading. So when, for example, I'm writing something here. Okay? I'm writing something. When I do a mistake, what I should do? I have to erase them and then correct the correct actually letter. This process known as a proofreading. Proofreading, it means when you are writing something, you are doing a mistake, you have to erase that and replace that with the correct letter or word. It's happening for the DNA polymerase. DNA polymerization is one of the most accurate processes in the world. 10 power to the 7 nucleotide, when is added by the DNA polymerase, just one mistake is occurring. 10 power to the 7, one mistake. But it's too much for DNA polymerization because we have a 6 actually billion. So this proofreading activity is added to DNA polymerase to decrease the rate of mistake. What's that? I, as a DNA polymerase, I'm adding the nucleotide. When I add one nucleotide, my direction is 5' to 3'. Okay? My direction is 5' to 3'. Then I have to go back. What is my direction? 3' to 5'. If it was correct, I'll actually left it and then continue. If it was wrong, I have to remove that and replace that, then I will continue. Each nucleotide which is added by DNA polymerase has to be rechecked one time. Then actually it can continue. This process by DNA polymerase is known as a proofreading and increase the accuracy 10 power to the 2 or 10 power to the 3. It means 10 power to the 7, increase 10 power to the 9. So it means the number of mistakes is decreasing to 10 power to the 9, just one mistake. So it's one of the most accurate processes in the world. So the last part is termination. So we talked about initiation, elongation, and then right now we have termination. Termination is a process like, maybe you saw, when you are driving in the highway, when you reach to the end of highway, what's happening? There are many actually signs, and also they create some stopping places to you decrease your speed. They give, yeah, stop, no, stop, decrease the speed, all of it. It's happening in our DNA also. At the end of the replication, we have a two sign. One of them is a protein which they are known as a TUS, terminus utilization substance. And also we have some sequence or site on DNA, replication termination. So we have a two fork. Imagine this is the prokaryotic, and here one fork is going from that side, one fork from that side. This side known as a clockwise. Clockwise. And this one known as a counter clockwise. So we have a clockwise fork. Thank you for nothing. Clockwise, counterclockwise fork. So for each of them, we have a specific site. Some of them, they are TER, termination replication. These counterclockwise, actually, they trap this. And that one, actually this one, they trap this. So why we add a lot of them? Because when this replication machinery pass from the one, the second one trap it. If they pass from the second, the third one trap that. Because maybe the speed is so high, some of the driver, they don't pay attention to the sign. They increase the speed, so we put a lot of these decreasing the speed. Then we can actually finish that termination. TUS and TER. It's so important to finishing the actually replication. So totally is the summary of DNA replication in the prokaryotic. Origin replication should be identified. Then RNA primer should be synthesized. DNA synthesis continues in leading. This continues in the lagging. Elongation in both strands is happening. And then we have to remove the piece of RNA, fill the gap by ligase. And finally, we have to organize them in the chromatin. So, first of all, when we are going to talk about the DNA polymerases in the prokaryotic, we have 5 kind of them. But these 2 are not important. What you guys say? What you need to understand from this slide. First of all, about the proofreading activity. Which one of the eukaryotic, prokaryotic DNA polymerase has a proofreading activity? All of them. All 3 kinds. DNA polymerase type 1, 2, and 3. About the 5' to 3' exonuclease activity. Which one has? Just one. For this reason, DNA polymerase type 1 is important for what? Removing what? primer. Because of this function. Let's have a look at the next one. We have a two process. Polymerization rate. Polymerization rate means nucleotide per second. Just pay attention here. It's so amazing for understanding what's happening. About the nucleotide per second. Type 1, 10 to 20. Type 2, 40. Type 3, how much? 1,000. What does that mean? It means when you are saying the one, just one second. DNA polymerase type 3. Add 1,000 of nucleotide and recheck all of them during that 1 second. Is it possible to understand that? No, it's not possible for us. 1,000 times adding and rechecking. It's so difficult. Because the time in molecular biology level is different from the time that we understand. If you are going to understand more about that differences, you can watch the Christopher Nolan movie. No, Oppenheimer. Interstellar, Inception, like that. These two movies, you can understand the meaning of the time in different areas. So in different areas, the time meaning is different. So you can understand that by watching this movie, the level of differences between these two areas. Exactly. Yes, I saw all the Christopher Nolan movies. So, Prestige is one of the actually most strong movie, but nobody don't pay attention to that because it's the one of the historical movie about that. You should know the philosophy of Greece to understand the Prestige. What's that? So, the second one, guys, finish the discussion. Processivity. Processivity, what does that mean? It means how many nucleotide is added before polymerase dissociate. What does that mean? It means how many nucleotide is added before dissociation of the enzyme. How much is added?
500,000.
So for this reason we are saying the DNA polymerase type 3 is the main polymerase in the polymerization. Because the speed of polymerization and the processivity rate is more important, is more than others. So what is the function of type 1? I told you. Tell me, please. Type 1 function. DNA polymerase type 1 function. Removing primer. Removing primer. Type 2 is important in the repair. Type 3 is the main one. So let's go talk about the DNA synthesizing in the eukaryote. In the eukaryote, you will study embryology, I think. Okay? Alhamdulillah, you will study embryology. So it's good for me, you are studying embryology because you can understand what's happening. In embryology, at the first day we are one cell. But what's happening? Division. How many hours does it take to do division?
Well, according to the phase, each phase have about 36
Totally
minutes.
in mean, actually.
You can say 3 to 4 hours.
No, 3 hours is not enough for doing a dividing.
7 days?
6 hours.
1 day. 1 day is 24 hours. We need 24 hours to do subdivision. Who told you just 6 to 8 hours?
It was written in our books that each phase takes about 36
Yes.
minutes.
the beginning they know, but the totally we need 24 hours for doing a cell replication.
Replication, I thought it was replication.
Cell division, cell replication is similar. Cell division, cell replication. It means replicate the cell. So, cell division or cell cycling consists of how many phases?
4.
What they are? What they are? G1 or gap 1 is in the higher school. Cell cycle. In the biology of higher school. It's not possible. It's in the higher school. Maybe in the elementary school also. Cell cycle is consists of G1, S, G2 and mitosis.
Take it for more information.
No, just that. So the cell cycling is consists of these four steps: G1, S, G2 and M. It's so easy. I think you study that in elementary or high school. No, you don't. But in the embryology also you study that. In the histology maybe you study that. Maybe you don't study anything. So any phase is consists of a time. Gap 1 is about 12 hours. But DNA synthesis 6 to 8 hours. 6 to 8 hours. We need to do a cell replication, DNA synthesis, sorry. So 6 to 8 hours. What is the time of cell replication in the prokaryote, in the bacteria?
18 hours?
Half an hour. 20 minutes to half an hour. 20 minutes. Yes, is correct. 20 minutes. 30 minutes.
I thought you were saying that you can have a 20-minute rest.
Rest? No, I didn't get. I think is it correct? I told you yes, correct. Just 2 minutes and then I will give you rest. Bacteria, you interrupt my focus. Bacteria cell cycle is 20 to 30 minutes. Eukaryotic is 6 to 8 hours. Eukaryotic genome is a billion. Prokaryotic is million. How much is bigger than prokaryotic? 1,000. So if the speed of cell replication in the eukaryotic be equal to the prokaryotic, at least we need how much? 20,000 minutes to 30,000 minutes. Okay? If we divide that to the 60 minutes per hour, is equal to how much? Near to 500 hours. So instead of 24 hours, we need 500 hours for cell replication. What does that mean? It means the time of pregnancy from 9 months should be 24 times increasing. Is equal to more than 12 years, 13 years. Just take for pregnancy. But it's not real in our body. It's just less than 9 years. How it's possible? Think about that, and then after the rest, you will tell me the answer of this question. How the cell replication, DNA synthesis, just during the 6 to 8 hours is occurring. When we compare to bacteria, it should be more than 500 hours. But during the 6 hours, we finish that. And you are going to make a new city, for example. This city consists of many buildings, okay? You have 2 ways One way is that you are starting from one point, start actually and build one building, then you are going to the next one. Then when it's finished, going to the next one. So how many time is taking? More than 100 years to finish the city. But imagine you will bring a lot of worker, a lot of engineer, a lot of actually equipment and material, and you will start from all the site at the same time, all the building, you know, like our brother Chinese they are doing during their works. When they are going to make some things, 2 days you can see they make actually a hospital, 5 days like that. So it's happening in the eukaryotes. So they are not starting from one site. They started from the many sites at the same time. Many DNA polymerases, many actually factors are working together, and they can make a DNA as actually in the short time. So we have multiple origins instead of one origin. So in the bacteria just we have one origin, but here we have multiple origins. So we have another actually differences, but in this slide actually we can compare them. Please answer this question to me, then I will be satisfied from my teaching. First of all, the origin of recognition in the prokaryotic, which molecule was responsible for doing that? In the prokaryotic, which protein recognized the origin? In the prokaryotic, that table I told you, the first molecule, the first protein, the name was?
DNA.
DNA. Thank you. DNA. Who answered the question? Thank you. DNA in the prokaryote. But in eukaryotic we have ORC, origin recognition complex. It's a complex of many proteins. So about the helicase activity in prokaryotes, what was? Yeah. What was the name of helicase in the prokaryote? DNA? B. Go ahead. What is in the eukaryotic? MCM. What? Eukaryotic cells. Eukaryotic means our cell, human cell, for example. Single-stranded protein in the prokaryotic they were SSBP. In eukaryotic is RPA. For primer synthesizing in prokaryotic we need primase or DNA G. In eukaryotic we need polymerase alpha. Primer removal in prokaryotic. In prokaryotic primer removal. DNA polymerase. DNA polymerase 1. Thank you. DNA polymerase step 1, removing a primer. In eukaryotic, RNase H, FEN. If you go back and look at previous question from previous semester, always I have a question from this table. And you should know which protein is equal to which actually protein in eukaryotic and prokaryotic and function. The last one, a sliding plan. What's that? You saw zip line. Okay. What's that? Someone bind to the zip and actually during the life slide on the zip. Imagine this is the DNA strand and the enzyme actually is bind to this strand. If bond, actually, a bond between these to be so tight, DNA polymerase cannot move. It should be actually like a zip line sliding on the strand, on the DNA. So we need a sliding clamp, like a zip line. This DNA should bind to the strand and slide on the DNA. This sliding clamp in the prokaryotic is the beta subunit of DNA polymerase type 3. But in the eukaryotic, known as a PCNA. Inshallah, in the first semester, when you study the immunology, you will study about some diseases like rheumatoid arthritis, lupus, erythematous, other kind of autoimmune disease. On those autoimmune diseases, you are actually studying the anti-PCNA again. And remember that, the PCNA, what was the PCNA? So the last part of the DNA in eukaryotic is telomere. So what is the telomere? Mer means member. Mer means member. Tele means far. A far member. Centromere means the center part. The center part. Telomere means the far part. Those they are so far. What's that? Actually, during the replication, because of problem of primer removing, all the time, when we are doing a replication, we lose some part of DNA from that side, from that side. Then when we are doing the next replication, we lose that part, we lose that. If we lose the important gene, the cell function or activity will be actually lost. Do you have any notebook for actually writing something? Yes, like that. Thank you. That is enough. Is our, for example, genome. So in this genome, there are many information in our genome. For example, is information for one gene. Other ways, there are information from the other gene. When you are actually bringing it to the university day by day, it's going to destroy. After many days, actually you lose this information, which is so important for final exam. And if you don't have this information for final exam, you will be failed in the final exam. The cell will be die. What's happened? They actually decide to add a cover, exactly. This cover protect from our information. Do we have any information on the cover? No, it doesn't important. Actually, we have information here, but imagine there is no any information. There is no any information in the cover. Just we added this cover to protect this value information for us. So what's happening? Imagine this cover also. Inshallah, after 7 years when you graduated, look at this. Also the cover will destroy. Okay? But
I have it from 3 years.
for 2 years, yeah, it can be actually continuing. But for 7 years, for 10 years, it will be destroyed. So these cover are telomere. What does that mean? It means we adding the cover to this DNA to protect our information. So during the whole history, from the beginning day of the human life till now, all human, they are searching about the elixir of life. What's that? Elixir hayat. Elixir of life. What's that? Elixir hayat. Immortality. Immortality. Immortality means we can alive to the forever, to the end of the universe. So immortality is a process which all the time, all the kings, all the actually powerful men, they search about that. Is it possible for us to be immortal? Is it possible?
No, it's not possible. Well, not yet.
Not yet. Yeah?
It will never be possible because of cell aging. In our DNA, the DNA in itself is programmed to age.
So if we go to talk about the science, we will have some problem in religion. Some people in religion, Christian, they believe Isa is alive. Shia still actually they believe that Imam Zaman is alive. Some people, all of the religion. But in biology, in science, as you mentioned, is it possible? No, because of, you said, what's that? Cell aging. Cell aging or senescence, oldness, is the process which happening in all organisms, in our body also. And one of the main process of cell aging is telomeres. Because by doing a cell replication, 1 time, 2 times, 3 times, 50 times, then we lose our information and cell will die. So this process happens all the time. We cannot stop that. The process of aging and senescence actually, they are related to another process in our body. Just I have a good example for that. There is a movie, the name is In Time. Who saw that movie, In Time? You saw that. What's that? What is the topic of the movie? It's about what? Yes, explain in Arabic. No, it's wrong. No, it's not. In Time is about the movie. It's about the movie, actually. In this city, instead of, yes. Tell me. Yeah. Explain that. It's correct. Yeah. Just 1 minute. Actually, it doesn't matter. Maybe you made a mistake by the name of movie. Doesn't matter.
I remember I see
Yes.
maybe 4 years ago.
Yes. It's been on to 2011. The movie is about the time.
انه اكو شخص بالمدينه، هاي المدينه ما يتعاملوا بالفلوس، يتعاملوا بالوقت.
Exactly.
يعني تشتري شيء ما تدفع فلوس، تدفع من الوقت لان انت شايل ساعه جديده. من يخلص الوقت ما تدفع من الوقت.
Exactly. So there is a just time for money. When you are working, you receive money. When you are actually buying something, taking a bus, car, you will pay the time. The time is actually on your fist and on your hand, on your fist. And you actually, there is a rich man, 1,000 year, poor man, 2 hour, 4 hour leg. And just they grow up till 23. All of their face is 23. The biological actually phase stop at 23. But they can be alive. The movie idea is so great, actually the idea behind the movie. The organization of movie and actually the structure is so actually bad. It's not a good movie totally. But the idea is amazing. If we able to stop the biological actually cell division, we can stop the aging. But this process, actually, as I mentioned, all the time they searching about that. Right now, Elon Musk and his team are working on some process which can increase the age to 150 years life. I'm not sure about that they can be successful or no, but they are working. But he knows there is no way to increase stop the aging or senescence. Why? Because of this telomere. But when they find out there is an enzyme which synthesizes the telomere, okay, these telomeres are covered. They thought with themselves, if we be able to increase the number of telomeres, we can increase the duration of time or stop aging. They thought with themselves is the elixir of life, telomere and telomerase. But when they did experiment on the mice, on animal, what do you think? What they find out?
They died earlier.
Why?
Because
Cancer, tumor. Because when you allow the cell to do replication without control, it leads to the cancer. So, one of the most important cells which has a high level of telomerase activity are cancer cells, tumor cells. In addition to tumor cell, what kind of cell in our body they have high level of telomerase?
skin.
Maybe then, no. You can actually.
Red blood cell.
What?
Red blood cell.
Red blood cell? No.
Yeah, they died like every 100.
A stem cell,
Yes.
germ cell. Germ cell, a stem cell, because they can do a large amount of replication. So these cells, they have high level of telomerase activity. And this telomerase, always when telomere is decreasing, when we lose the cover, added a new cover. So cell can continue.
So, Professor, is there a way to find a technique that slow the aging of the cell without using the telomere?
We have many actually theory about the aging. One of the theories is aging, one of the main of them. They are working about the other. For example, one of the theories belong to the mitochondria. They are saying everything is based on the mitochondrial function. One of the theories is based on the function of the cardia. They are saying the cardia is the main one for the aging. But one of the theories, just one of the theories, is about the telomere. So they thought with themselves, if we are not able to increase the telomerase activity because lead to the cancer, we can look at that from the other side. If we be able to inhibit telomerase activity in the tumor cell, we can treat cancer. So right now they are working on some drug which they are inhibiting of the telomerase function, and they are in the clinical trial phase 3. Maybe in the next year they introduce this drug to the market, and they are so good drug for the anti-cancer. So what is the telomerase? Telomerase is an enzyme, actually. It's the enzyme which consists of two parts. Is a mitotic clock. Mitotic clock is a clock of mitose. It means when we do a one replication, the clock of our life is going to the end. And finally, when reach to the end, our life also is ending. For two processes, aging and cancer is so important in telomere. Telomerase is an enzyme. How it can add the cover? By these sequence, because it has its own sequence. If you look at that, on telomerase, we have a two actually part. TRT, which is the protein part, and TRC, which is the RNA sequence. By this sequence of RNA, telomerase can add this telomere to the end of the DNA. 6 actually nucleotides, A, G, 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 T, T. Maybe 1,000 of these hexamer is added by telomerase. 1,000 of this hexamer is adding, and then telomerase actually do its function very well. So prevent from the shortness of the DNA. As I mentioned, it consists of two parts. TRT, TRC. TRT is the protein part, TRC is the RNA part. What is the function? Synthesizing of telomere. What is the activity? Is reverse transcriptase. What does that mean, reverse transcriptase?
The opposite of transcriptase.
Exactly. What does that mean? Making DNA from RNA. Because it has an RNA, convert that RNA to the DNA. For this reason, it has a reverse transcriptase activity. It's active in what kind of cell?
Germ cells, stem cells, and cancer cells.
Cancers. In these three kinds of cells is active. What can be the benefit of useness of
Treating cancer.
For treating cancer, we can use the inhibitor of the telomeres. So is the whole idea behind the telomeres. So related to the last slide for the replication just, we have some drugs, they can act as an antibacterial agent. Some drugs, they can act as an anti-cancer agent. Ciprofloxacin, nalidixic acid, and novobiocin. All of them, they are inhibitor of DNA gyrase. Is the name. Do, it means doing the similar function. Ciprofloxacin, nalidixic acid, and novobiocin are inhibitor of DNA gyrase or topoisomerase. In anti-cancer, etoposide, adriamycin, and doxorubicin, they are inhibitor of human topoisomerase. Do the same. Human topoisomerase inhibitor. Etoposide, adriamycin, and doxorubicin. We have another anti-cancer or chemotherapy agent, which the name is 6-mercaptopurine, which is so famous. It's inhibitor of human DNA polymerase. And also we have a 5-fluorouracil, which is the inhibitor of the thymidine synthase. I forgot one point about the human DNA polymerase. Instead of type 1, type 2, type 3, we have DNA polymerase alpha, beta, delta, epsilon, and gamma. What is their function? Alpha is equal to the primase, as I mentioned. Beta is similar to the DNA polymerase type 2. Is important in repair. Delta and epsilon, they are equal to DNA polymerase type 3. Delta for leading strands, epsilon for lagging strands. Leading strand and lagging strand. And gamma is a mitochondrial DNA polymerase. Mitochondrial DNA polymerase. If you wanted to actually, in this slide I brought them, but I passed them. DNA polymerase alpha, delta, epsilon, beta, and gamma. Gamma is gold side, but it's not clear here. So simple that. What's happened, guys, in the classes?
Based on the gene of electrons basic go on.
Also is correct, but it's not the simple definition. It's so simple that definition.
Mistakes in the
Yes. We can say any, yeah?
I would say it's a change in the location of the nucleotides in a gene.
Any change is known as a mutation.
Exactly, good or bad. Whenever reproduction happens,
We have another condition in addition to change.
Unrepaired.
What?
Unrepaired.
Exactly, thank you.
They can't
Any unrepaired error or mistake or damage, unrepaired error, mistake, error or damage known as a mutation. What?
Going back 3 to 5 direction.
No, doesn't matter. If we lose, if we change one nucleotide also, it's known as a mutation. So, imagine you are going to the city, to the new city. Which city in the Iraq has a high level of accident? Baghdad. Imagine you are going to Baghdad and you are seeing all the cars, they are crashed. What is the reason?
One car.
The reason is women.
You can't drive to save your life.
It's not actually a good answer.
Yes,
It is a reality.
You know? Let me to actually explain 1 minute for you. We are living in the Middle East. Living in the Middle East, actually, itself is disaster. Itself, living in the Middle East is disaster. And because of our religion and our ancient and history, the women, they are living in disaster 2 times, 4 times than men. Okay? So you, as a new generation, as an educated generation, you shouldn't actually doing the same thing like your father, like your grandfather, like the previous. I know it's a kidding, actually it's a joking. You don't have actually intention to do that. But when you actually simplify that, it can be actually so important in the total actually social, public, in our population, everything. So we should correct ourselves about that. Maybe it's just a joking, it's a kidding actually, a simple kidding, but totally has a side effect. Yes, you're right, some women, they are not a good driver. But some women, they are actually good driver than the men. We have men also, they are not a good driver. But maybe you think totally the women are not good as men. Why? Because they didn't drive more than men. Okay? We have to give them this chance. Maybe someone actually is not a good driver, but you need to help them. So do not actually focus on this kind of thinking, not just about the driving, about everything. It was actually not related to my discussion, but I wanted to tell you, pay attention more about what you are saying, what you are telling in the class, especially in the public. But let's go back to our question. What was the actually high crash car?
Because high amount of population.
High amount of population is one of the reasons. Then traffic light. Yeah, there is no good traffic light. Then
So this organization is very well. So
exactly, the organization is not good. Actually, they can't handle them, guidance them very well. Maybe the driver are not careful about the driving. Road are not good. Yeah, exactly. The weather maybe. Yeah. This reason, yeah.
It's a sequence. Like if someone has a car crash, so the other cars will involve.
Yeah, maybe. Totally, all of these reasons together, we can say the rate of accident is high. The rate of accident is high. It's maybe due to the traffic light, it's maybe to the police officer is maybe due to the car itself, quality of car, maybe due to the weather, maybe due to the road. All of them together, they lead to the high rate of accident. The other reason is that we don't have enough mechanic, a fixer, to fix that crashed car. So it can be divided to two categories. First, the rate of accident is high. Second, the rate of fixation is low. So imagine in our cell what's happening. In our cell also we have accident. A lot of accident. A lot of mutation. Day by day, second by second. So imagine we have this amount of accident, this amount of fixation. So they are in balance all the time. What's happening when the rate of accident is increased? So the mutation is increased, right? Otherwise, imagine the rate of fixation is decreased. What's happening again? Mutation is increased. So mutation can occur due to two reasons. The rate of accident is increasing. Why the rate of accident is increasing in our body? When you expose to those compounds, to those actually conditions which they increase the rate of mutation, like what? UV radiation, X-ray radiation, chemical compound, nitrous, actually nitrate, or other kind of toxin which they are using in the agriculture. Air pollution, unhealthy water, all of them, they increase the rate of mutation. Otherwise, a lot of mutation, maybe they can become actually physical compound, chemical compound, different. About the fixation also, we have a different system in our body to prevent from this mutation. This actually system also due to the problem, maybe unhealthy food. One of the reasons of mutation is that the fast food, actually. Because this fast food and the kind of cooking the fast food also is dangerous. But why mutation is important? Because the mutation leads to the disease. What kind of disease? Cancer. All of the cancer are due to the mutation. So we need to actually know about the mutation. If we go, actually, we define the mutation. Mutation is unrepaired damage, unrepaired error, unrepaired mistake. What are the reasons? Spontaneous, loss, losing, and alteration of this. Chemical, nitrous acid, radiation, UV, ionizing. And also during the replication. During the replication, do we have a large amount of mutation or is low? How much mistake we have during the mutation, the replication? I told you. How many mistakes we have? We had a 6 multiplied by 10 power to the 9 nucleotides. How many mistakes is happening instead of 10 power to the 9? 1 mistake. So how many mistakes we have totally? 6. 6 mistakes happening during the 1 replication. Is it too much?
Yes.
Why?
Because the other one will depend on this. So it will repeat the mistake, something like that. The mistake will repeat it.
Can you explain more? Yeah?
If a single nucleotide gets changed, everything changes.
Maybe, but most of the time there is no any change in our body by changing in the.
It depends. Sometimes.
Yeah, it depends exactly.
Disease, sometimes.
The damage will be replicated from one from one DNA to another, so it will cause genetic damages and genetic diseases that will be transferred from one generation to another.
Yes, some of what you are talking about, yeah.
Example,
Let's look at the number. 6 mistakes in one replication. In one cell. How many cells we have?
Billions.
10 power to the 14th cell we have in our body. Some of these cells, they are not doing replication during the year. Maybe during the 10 year they are not doing the replication. Some of them, every day they are doing the replication. Some of them maybe one time per week, per month, they are doing the replication. It's different. But totally, if you imagine, during a 1 year, each cell do a 1 replication, it's just imagine, you know. We have how many mistakes in 1 year in our cell? The number is not actually understandable. If you are going to understand what's the number, if we do it per year, okay? If we are going to calculate for 2 hours, is equal to the what does that mean? It means during that 2 hours you were in the class, near to 60 billion mistake and error happen in your body. Should we worry about that? No. Why?
Because it's normal.
Because when we study the transaction, there is a lot of polymers and a lot of techniques to repair every damage.
Exactly, because of our repair system is a powerful system which doesn't allow this actually mistake happening in our body.
It is rarely happening.
So the last actually question and the last part of my teaching for today. The mutation all the time are hazard and dangerous, or sometimes they are.
No, it's adaptive.
So yeah.
For example, eye color is a mutation, came from a mutation. Lots of our characteristics like phenotypes come from mutation.
Exactly. Totally. If you are going to simplify that, what's that?
As long as the humans progress, or not humans, and
Any organism doesn't matter. Yeah.
The more reproduction they have, like the new generations will have new mutations.
We are saying what? Evolution. So evolution and natural selection is based on the mutation. How? These mutations, some of these mutations, give new characteristics to the some species. So these species, they can be alive and adapted to the new environment, to the new condition. So always is not dangerous. Sometimes it's so benefit actually. It's so beneficial for the adaptation to the new condition. It's amazing in the actually biotechnology how we can use these mutations to create a new drug, a new antibody against a disease. By just doing a mutation in some protein, in some antibody, we can create a new antibody which can treat some disease. So always is not dangerous, can be also useful, especially for the evolution, not revolution. Revolution means another actually. Yeah, evolution or development is so important. So if be alive for the next week, inshallah, on Saturday I have another classes, and on the Sunday we have a practical session. I like movie too much, yeah. There is a movie, the name is, the last movie of DiCaprio, what was the name? One Battle After Another. If you have a time, see that, because it's related to the extraction of DNA. And in the next week, I'm going to. Who saw that movie, One Battle? It's about the extraction of DNA. Do you remember that part?